Hello, my name is Renee Hallstein. I'm a research assistant in Dr. Linda Lee's Chem Lab. I'm involved in the PFAS fate and transport in a field study with land-applied biosolids. During the summer of 2023 and the 23 through 24 school year, I worked as a research assistant for Linda Peter in Lee Lab on Purdue campus. This environmental fate lab conducts research into PFAS and UOC contamination of groundwater, soil, and biosolids for environmental impact and human health. The study examined soils from a farm in Virginia that uses biosolid application for nutrient release. The soils on the farm are Atlantic coastal plain with low moisture and nutrient retention. This is why biosolids are used. Soils with applied biosolids can have PFAS transport. The study aims to understand PFAS fate and movement in soil, groundwater, and surface runoff. Corn, soybeans, and wheat for animal feed are grown. 3 soil types are present at the farm. A slowly permeable acre-dill silt loam, shown on the map in purple on the left side, a deep and moderately permeable Bojack sandy loam, shown in the reddish-orange to the right of the map, and its motley loam, shown in the green and runs through the middle of the other 2 soil samples. Now, the main part of this experiment I handled was the physical extraction. The first part of this was grinding the soils that we received from the farms. In order to grind them, we had to dry them down and weigh them. The next step was some general prep. This included labeling and weighing the empty tubes and the injection vials. Next, we weighed 0.5 grams of the soil into 15 milliliter tubes. For each soil, there were 5 tubes in total. 3 replicates, and 2 controls. The controls had the natives, while an internal standard was added to all 5 of the samples. Next was the actual extraction. First, we started by adding 7 milliliters of a methanol and ammonium hydroxide mixture to the 15 milliliter tubes containing the soils. Next, we vortexed for 20 to 30 seconds, shown in the first photo on the left. Then we sonicated for 60 minutes at 30 degrees Celsius, shown in the second photo. And then we centrifuged for 30 minutes at 3,000 RPMs, shown in the third photo on the right. The next step was to decant the fluid into 50 milliliter tubes. Then we repeated these steps, starting with the addition of the 7 milliliter of methanol and ammonium hydroxide solution. In total, we repeated 3 times, and we ended up with about 21 milliliters of fluid. Now, this solution was dried down in the rapid vap to about 0.5 milliliters to receive a concentrated solution of methanol and ammonium hydroxide mixture with the PFAS particles. The next step in this extraction process is to prep for injection into the LCMS. We did so by prepping mini centrifuge tubes, transferring that 0.5 milliliter of the concentrated fluid into them, and then another centrifuge. Now, for the actual sampling technique of this experiment, biosolids are sampled before application. Soil samples are collected before, immediately after biosolid application, and post-harvest. So in total, we received 3 different sets of the same 3 soils. The soil samples go through the extraction method and then are injected into LCMS. This is a liquid chromatography mass spectrometry machine. It reads the retention time and weights of the molecules. Once several sets of the injection were completed, we analyzed the results. The replicates, 1, 2, and 3, are compared to the MC and MCSD. These are the 2 controls that have the native. We look at the retention time, the height of the peak at the retention time, and the total weight of the molecule. This analysis is done for several classes of PFAS. PFAS migration from the soil surface is known to occur on soils receiving biosolid applications. However, it is less understood what soil qualities affect this transport. This study focuses on 3 types of soils: identifying the differences in PFAS transport between them. The data from the study will identify soil characteristics that are suitable for receiving biosolid application. This allows for the development of site-specific strategies for the beneficial use of biosolids. Thank you.